Welcome, Brookings Bio students. This is Mrs. Rydell, and we're going to look a little closer at part of that cell cycle that we looked at in our last vodcast. If you remember, in our last beginning of our PowerPoint, we looked at cell cycle and we looked at the phases of interphase, G1, S, and G2. And we talked about mitosis a little bit, but we didn't really go into depth about what is happening in those phases. Remember, we wrote out the different things that were going on in each of the parts of interphase in our little pie diagram. And today we're going to look a little more closely at those mitosis phases. So if you remember from the last we talked, interphase is made up of G1, S, and G2 phases. And interphase is the in-between dividing phase. It's part of the cycle when the cell is not dividing. It's hanging out in the body, doing its job. And cells are in interphase most of their lifetime. If I look at an interphase cell under the microscope, I can see a nucleus. I can see a nucleolus, that dark spot where we're making ribosomal RNA. The DNA is spread out as chromatin. I don't see individual chromosomes. It looks very grainy. And that's because we have the DNA open in a form that we can read the directions, read the information, and know what to do. Remember, in interphase now is where the cell is going to make the division or the decision to divide or not divide. And if we are going on to do cell division, we're going to copy our DNA in S phase and make all those cell parts and molecules we need for the new cell in G2, and then we're going to go into prophase, first dividing phase. The way I remember it is pro has to do with, think professional sports. When you think about sports, you have minor leagues and the pros. If in baseball, professionals are the number one best players, and prophase is the number one very first dividing phase. So what we're going to do in prophase is set up the cell now, get out the machinery that we need to actually move the chromosomes around. So one of the things we need to do to move our DNA is to make it transportable, scrunch it into chromosomes. Remember, we talked about taking your homework home in your backpack. If you pack it up, it's easy to move. But once you get home, if you want to read the book, if you want to do your homework, you have to open things up. You have to pull it out of the bag, open the books, read the information, and then when you want to bring it back to school again, you close the books, you pack it up. It makes it easy to move, and that's what the cell does with its DNA. It takes that chromatin, that loosely packed material, and scrunches it tightly into chromosomes. The other thing that we need to do if we're going to move chromosomes is we have to get up the apparatus that's going to help to move those chromosomes. And remember, from our cell parts chapter, we talked about centrioles in animal cells. The centrosome area is going to help to organize our spindle. Those are the microtubule fibers that are going to move our chromosomes. We need to get rid of our nuclear membrane because nucleus is containing the DNA, remember, in a eukaryote. And if I want to move my chromosomes around inside the cell, I have to let them loose, get them out of the nucleus so they can move. And the last thing I need to do is to set up that spindle so that the chromosomes have attachment place in order to move them. And all of those things take place in prophase. It seems pretty complicated and seems like a lot to know, but it's once you get this phase down, the other phases are pretty simple because we have some clue words to help you remember what's going to happen in the other phases. If you remember now, this is a new vocab word. We talked about centrioles before in our animal cell when we talked about cell parts. The centrosome is a region in the cell, an area that's organizing this spindle. Remember, the spindle fibers are microtubules that are going to attach to the chromosomes and help to get them to opposite ends of the cell during mitosis. And the centrioles in an animal cell sit in that centrosome area. Okay, so we're ready to go. We've made our DNA copies. We've made all the cell parts we need. We've gotten out our machinery to move the chromosomes. And so the first thing we do is to line them up in the middle of the cell. M for meta, M for middle. The cells line up in the middle. And that's so the cell can check to make sure each chromosome is attached to a string. If you remember that Home Alone movie, the very first movie, and they wake up late and they're rushing to the airport, and the mom says, go down to the car and line up everybody and count heads to make sure that nobody gets left behind. Of course, we know somebody does get left behind, but lining people up and counting is a way to make sure everybody's ready to go, everybody's attached. And so that's what the cell is doing, lining up all the chromosomes in the middle of the cell so that it can check to see, is everything ready? Because if one of the chromosomes doesn't get attached to a spindle fiber, when the chromosomes move, that one's going to get left behind, and that means the cell would be missing that chromosome if one of the cells is not going to get that chromosome. And that would be very bad for the cell. It's not going to be able to survive if it doesn't have a copy of everything. Next phase is anaphase, and A for ana, A for apart. In this phase, the chromatids are going to separate. Remember, our chromosome is made up of two chromatid arms that are identical copies, held together by its centromere in the middle. And now we're going to split the copies up. Centromere will split. Each chromatid is going in opposite direction. They're going to go apart, and it makes it so each of the daughter cells now is going to get a copy, one copy of each of the chromosomes. They're going to have all the DNA that they need. The third phase is called telophase, and T for telo, T for two. When I can see two little bundles of DNA in the cell, I'm in telophase. Telophase is sometimes called reverse prophase because everything that we did in prophase to set up the cell for division, we're going to undo now to turn it back into an interphase cell. So if you remember in prophase, we lost our nuclear membrane. Now that has to come back again. The cell's going back into interphase. Our nucleolus starts making RNA for proteins to make ribosomes that are going to make our proteins, and nuclear membrane comes back to contain our DNA. So we're resetting the cell again. One of the other things that happened in prophase is we took our DNA and scrunched it up into chromosomes so that's easy to move. Now the cell's going back to doing its going back into interphase and doing its job. So we need to spread those chromosomes back out as chromatin, open up our information so we know the directions of what to do. Centrioles and spindle are going to disappear. We've done our dividing. We don't need them anymore, and they go back, won't appear again until we're ready for our next cell division. So we're kind of undoing everything that we did in prophase by reverse prophase, doing telophase. So far we've divided up the DNA, that's mitosis, but we still have one last thing to do, and that's to divide up all the cell parts. Each of the new cells now needs some of those cell parts that we learned about in our cell parts chapter. So the last phase is cytokinesis, split the cytoplasm into two cells. And animals do that by pinching in the middle. They make what's called a cleavage furrow. And the sides of the cell pinch in and keep pinching, squeezing, and break into, and I get to what we call daughter cells. Plant cells, though, have a little problem because remember, plant cells have a very sturdy cell wall on the outside. We talked about cellulose making plants sturdy. Cell wall prevents plant cells from doing that pinch. They can't make a cleavage furrow. So how am I going to separate my cells? I actually have to build a new little wall in the middle. You can see the wall starting to form in between the two nuclei, and here's my cell. And so plants actually build a new little wall. It's called a cell plate. And the cell plate is going to make a dividing wall now between the two sides of the cell and divide that cell into two cells. And then now this wall can expand a little bit as the cell grows, and we get the cell can still grow bigger, but that little dividing wall is going to separate. Connect to something you already know now. Remember in our cell parts chapter, we learned about a cell part acts like the UPS of our cell, post office, to package molecules up and ship them somewhere. And we talked about shipping out of the cell, but another place that can ship is somewhere in the cell that needs material. And in this case, our Golgi bodies now are going to package the material we need to build this new wall and ship it to the center of the cell and dump it there to start building the wall. So Golgi bodies help to build the cell plate during mitosis. Okay, so think about our phases now. Remember, this is a series of steps, a sequence. If you try to think of this as just random, I got to memorize this phase, it's complicated, and it seems like it doesn't make any sense. But if you think of it as a sequence, kind of like a movie, and this has to happen before this has to happen. Before I can pull my chromosomes apart, I got to get out my spindle. Before I put my nucleus back, I have to have split the chromosomes up. So think about it as a sequence, and it makes more sense. So think about the phases we've talked about. Interphase, hanging out, doing our job as a body cell. Next phase is prophase, copy or get out the machinery we need for cell division. The spindle appears, centrosome area organizes that spindle. We lose our nucleus, chromosomes condense to make them easy to transport. Line them up in the middle to make sure all the chromosomes are attached and ready to go. Apart, we pull the copies apart so that each daughter cell is going to get one set of chromosomes, and then split our cytoplasm. Split our DNA into two cells. We reset the cell by bringing our nuclear membrane back and getting rid of our spindle fibers and our centrioles, and then finally cytokinesis splits the cytoplasm, and we have two new daughter cells. We're going to kind of make a little note sheet for ourselves about that. And before we do that, we're going to watch a little video about the phases.
Cell cycle includes all phases of a cell's life. For about 90% of the cell cycle, the cell is in interphase. During this phase, the cell grows and copies its chromosomes. The chromosomes exist as long, thin strands of DNA within the nuclear envelope and nucleolus. Two centrioles, a single pair, lie at right angles to each other just outside the nucleus. Near the end of interphase, the cell prepares to divide. At this time, the DNA is replicated, producing identical strands called sister chromatids. The sister chromatids of each chromosome are attached to one another at the centromere. The centrioles are also duplicated. Now the division of the cell nucleus into two identical daughter nuclei begins. This process is called mitosis. The first phase of mitosis is prophase. During prophase, the replicated chromosomes condense. Sister chromatids remain attached at the centromere. The two centriole pairs move to opposite sides of the cell. The nucleolus and the nuclear envelope dissolve. The mitotic spindle forms between the two centriole pairs. A spindle fiber attaches to the centromere on each chromosome. The spindle fibers begin to move the chromosomes to the center of the cell. The second phase of mitosis is metaphase. The chromosomes line up on a plane in the center of the mitotic spindle. The third phase of mitosis is anaphase. The centromere of each chromosome divides. The sister chromatids separate and are pulled toward opposite ends of the cell by the spindle fibers. The fourth and final stage of mitosis is telophase. Daughter nuclei form at each end of the cell. Mitosis is then complete, but the process of cell division continues.
Okay, so let's watch our next little part that has to do with cytokinesis. There we go.
During cytokinesis, the cell membrane begins to pinch the cytoplasm of the cell in half. Cytokinesis and the process of cell division is complete when two new daughter cells are formed. After cell division, the cells re-enter interphase and begin another cell cycle. The chromosomes uncoil, and the cell resumes normal activity.